And these are things we can look at or we can assay for, okay? And the observation was that there was a strain of corn that had an unusual chromosome structure. So a normal chromosome 9 should look like this, and you can see that in this case, the C and WX genes are linked along the same piece of DNA, along the same chromosome. And over here we have the homologous chromosome, but at this end we have this kind of funny little knob structure, and at this end we have a rearrangement where part of chromosome 8 got attached to the end of a normal chromosome 9. And it turns out you could actually see the knob, or you could see this chromosome 8 extension. Okay? So you could visualize something. These are cytological markers or microscopic markers that you could actually see. You can't see nucleotides in here, you can't see genes, but you could see big objects like this. And it turned out whenever there was a knob, it predicted this big C allele. And whenever there was this extension of chromosome 8 off of chromosome 9, that would predict this small WX allele of that WX gene. So these are cytological markers, and so they would predict what the genotype or what the phenotype would be through various matings and so forth. So this kind of experiment gives you an idea that chromosomes have something to do with traits and inheritance. Okay, it's, I mean, obviously kind of crude, but this is a starting point for people to realize what it is in the cell that's carrying information and transmitting information. And you know, a lot of studies after this showed obviously that it's DNA and nucleotide sequences and all that kind of stuff. So that's one interesting point about this. The other interesting point, which is relevant to our discussion today, is that sometimes the chromosomes would undergo a rearrangement or recombination event where now the knob here would be associated with another end of chromosome 9 that was normal, and this chromosome 8 extension was associated with the other end of chromosome 9, which is normal. So something had happened in between them to kind of swap information somewhere in the center to cause the ends to be rearranged. And so you could see a rearrangement or a recombination of chromosomes, and yet you could still make an accurate prediction about what the phenotype ought to be. Still, if you saw the knob, you would predict big C, and still if you saw this chromosome 8 extension, you would predict this allele small wx. Okay, so just a kind of simple early experiment giving us some idea that it's chromosomes that have something to do with inheritance. So that's one conclusion. The other conclusion is evidently these chromosomes can be rearranged or recombined. So great. Okay, we saw this before. I just want to kind of reiterate that you can take a metaphase spread of, let's say, human chromosomes from a human cell, and if you know how to do this, you can stain them and you can line them all up, and you can obtain a karyotype which is organized from chromosome 1, the largest, down to smaller chromosomes ending here at 22. And of course we get a paternal chromosome and a maternal chromosome, or vice versa, depending on which is which. So I just want to remind you that this is the chromosome structure of humans, and if there's 20,000 genes, we have to have 20,000 genes distributed amongst all these. And another reminder is that the amount of sequence in the human genome that actually encodes those 20,000 genes is pretty small, right? There's a lot of other kind of dark matter, junk DNA that we don't know what its function is. So the amount of gene DNA that you see here is a small fraction of the kind of total picture. Okay. So we're going to talk about linkage of genes, and I'm going to go through this in various ways and present various cases for what is true and what isn't true. But basically the idea is if every 20,000 gene unit in humans was completely distinct, you know, you could imagine they are all their own chromosome, and you have to split them up during mitosis and meiosis. That's a possibility, okay? So it's conceivable that there's 20,000 little chromosomes. That turns out not to be the case, but that's one kind of extreme example. In reality, there's a number of chromosomes, and all the genes have to be stuffed into those chromosomes, right? And that's going to vary from organism to organism depending on how many chromosomes they have, and so forth, but it means that chromosomes have genes that are linked along the length of the chromosomes, hundreds of them or thousands of them. Okay, and just a reminder again, that the chromosomes that you get from your mother will have all of your maternal derived alleles. The chromosome set that you get from your father contains all your paternally derived alleles. And we'll come back to this later when we look at adults as mixers of their own parents' DNA. Okay, so let's think about the distribution of alleles or genes during meiosis. Case one, it's possible that you have a single gene, comes in two allelic forms, big S and small s, and this is pretty simple, right? You know the chromosomes are going to separate, and so the gametes are either going to be big S or they're going to be small s. No big deal, that's kind of a basic precept. Case two, if we consider two genes, the Mendelian situation is such that whatever you get at your S gene is independent of what you get at the T gene. So if you get big S allele from S gene, this other chromosome is going to separate independently, and so you're going to get either big T or small t. If you get the small s, you either get big T or small t. So everything kind of proportions itself out into 25%, 25%, 25%, 25%, right? That's kind of the main Mendelian rule. Case three, here's a possibility that you have two alleles of this two genes, S and T, but they're linked. So you've got an S gene here, and you've got a T gene along the same chromosomal DNA. Question is, what happens then? And one possibility is that they always go together. So whatever you have on one strand, one chromosome, you're going to get the associated allele of the T gene because they're connected physically together. So if we suppose that big S allele is linked along the same piece of DNA to big T, then linkage would mean if you get big S, you always get big T, okay? Or if you get small s and small t is located nearby, then you would always get those together. That's the idea of linkage, okay? And that's partly true, okay? Case four, we will have recombination which separates the S and T genes once in a while at some frequency, and that frequency is important. We can use that to map genes along DNA and so forth. So this is the kind of thing we're thinking about, and the initial example we'll look at involves a couple of genes on the X chromosome in Drosophila. So we already know about this red eye, white eye system. Big W causes dye production and you get red eyes. Small w doesn't cause dye production, so you get white eyes. There's another linked gene on the X chromosome in Drosophila that we can look at the same time, which is big M for normal wings and small m, miniature, for miniature wings. And the genes are linked on the X chromosome, and that's kind of the puzzle that we're dealing with in today's lecture. The question is, how will eye color and wing phenotype genes be transmitted due to the fact that they're linked along the same piece of DNA? Flies have about 15,000 genes, so it takes about 15,000 genes to make this. It looks like he's standing in the doorway in a robe, inviting you into the library or something like that. These are great kind of pictures, so complicated, right? And you need 15,000 genes to make something like this. And here's just some more pictures of that type of thing, what we're talking about. These aren't necessarily Drosophila, but they're flies, right? Okay, so I've got case one. Case one here is some possibility that we might imagine happening. Let's suppose that we've got X big W and X big M on one chromosome, and we have X small w, X small m on the other chromosome. One possibility is that linkage is meaningless, doesn't matter at all. It's possible that the cell will just break everything up into 15 or 20,000 different units and then spit them out to the gametes during meiosis 1 and meiosis 2. So it's possible that linkage is irrelevant. It means nothing. And that if we had X big W, big M on one chromosome, those separate. Okay? And if that happened, then still it would be a Mendelian situation where X big W would combine independently with big M or small m, right? Because we're saying even though they're linked, you chop it up. So this is at least one theoretical possibility. If this were to happen, you'd get 50% recombinant, 50% parental phenotypes. It would be the same situation as if the W and M genes were on completely different chromosomes. So is this what happens? No, it's not. It's not what happens. It's kind of an extreme case. Okay, so pictures here, pictures here. Here's an artificial fly, right? People try to make these little robots, and even multiple robots, and get them to fly around together and so forth. It has little wires. So this is our best attempt to make a fly. And this is a really useful tool for flies. Okay, and the case here, the possibility is, let's suppose we start out with this same female heterozygote, big W, big M on one chromosome, small w, small m on the other chromosome, and let's suppose linkage was absolute. You could never separate genes if they were linked together by nucleotides. So that's a possibility. It's kind of the other extreme. Okay? If linkage was absolute, then if you got big W, you would always get big M, if that was the chromosomal arrangement. And if you got small w, you would always get small m. And this is a possibility. There's nothing wrong with this necessarily. The question is this how it works? Is this what happens? No, not exactly what happens. Okay? So what happens is kind of intermediate. Sometimes linked genes stay together. Sometimes they separate through recombination. Okay, so our possibility here is, again, we start out with a heterozygote female fly. If linked genes sometimes transmitted together, but were sometimes unlinked or separated by meiotic recombination, then usually you get a parental chromosome of big W, big M, or small w, small m. But once in a while, the offspring would have those two shuffled so that you get big W, small m, or small w, big M. So you'd have recombination. So now you get some fraction of the progeny being recombinant compared to some fraction being parental. Okay, so in this case, you get some percent parental, which is either this or this, and some percent recombinant, which is this or this. Okay, so is this what happens? Yes, that's what happens. And the question here, which you've already read, I'm sure, is this what happens when you accidentally enter a matter exchange transporter device along with the fly and come out a while later? Clearly, that's what happens. Okay, so that's part man, part fly. Okay, so we'll set the experiment up with some pictures from the book. We start out with the female true breeding line, which is homozygous recessive for W and M, made it to a wild type fly, male fly, and we get the F1 heterozygotes. The reason I guess the book starts with this is typically maybe in a laboratory you would start out with your pure breeding, true breeding strains, and then you set that up to generate the offspring that you want to do in your actual experiment. So the first stage of this is we're trying to generate a female fly that has two X chromosomes, but she's heterozygous for two genes on those X chromosomes. So this female fly should be X, W plus M plus on one chromosome, and X small w, small m on the other chromosome. That's what we're trying to achieve here. And then we're going to ask the question, are we going to have recombination or separation of the W and M alleles in this female? So that's kind of part one. Part two will be to cross the F1s we generated in the previous experiment, that's highlighted again here, and obtain F2 progeny and ask the question, well, what do we get, right? So one extreme would be only parental type progeny. The other extreme would be 50% parental, 50% recombinant progeny. And the kind of middle ground, what really happens is you get a certain percentage that are parental and a less than 50% of the progeny that are going to be recombinant. So if we kind of look at a set of offspring that might be produced from a cross like this set over here are parental varieties. And what parental means, sometimes it's confusing, but what parental means is that the connection between the type of wing and the type of eye that you see in these four offspring classes are the same as what you have seen in the parentals. Okay, so that's the parental type. Recombinant means that you have mixed and matched the W and M alleles to generate new varieties or new combinations of alleles at these W and M genes. And those recombinant progeny are shown over here. Okay, And in this particular experiment, they're suggesting that we get, 1,500 parental types, which are going to split themselves into males and females, right? Because this is an X-linked example, so we have to deal with this kind of sex ratios. And we get 900 or so recombinant types, total numbers of recombinants. And this gives us information about how close the genes are, or how far apart the genes are, W and M. So something like this is kind of routine. It happens. You get progeny in these types of numbers. This is a different process than something like meiotic non-disjunction. Meiotic non-disjunction, in contrast, is extremely rare, right? It's a kind of accidental event that you might see in the background of otherwise completely normal offspring, but it would be extremely rare. This isn't something that's extremely rare. This happens routinely as a way to deliberately break apart maternal and paternal chromosomes and shuffle the alleles to generate new types of new allele combinations. Okay, so pretty straightforward. Here's what's happening in some picture form. I use this big W, big M nomenclature because it's sometimes a headache to put all those superscripts. But this is supposed to be red-eyed and normal wing. That would be one chromosome in the female that we're talking about. The other chromosome is small w, white-eyed, and the miniature wing, small m. So we start with the female who is heterozygous, like this, and it might be X-paternal, X-maternal, or switched, it doesn't matter. But we start with this, we replicate, so we get these chromosomes lined up in the synaptonemal complex, and here is, I'm just depicting one possible recombination event where we cross over in between the W and M genes to generate recombinant chromosomes. Some of them are still parentals here and here, but the two internal ones in this particular example, we've now shuffled or switched the arrangements that we had previously at the W and M genes. So these would be recombinant haploid gametes available for fertilization and so forth. So this is basically what's happening at a certain frequency in this female fly, okay? Now, similar types of things may be happening in the male as well. We have to remember that there's an X and Y here, but we're not visualizing anything that's happening here. This is the heterozygote. This is the one that we're actually going to be visualizing in the experiment, where we're going to see this shuffling of dominant and recessive alleles. Also, the system here is an X-linked system, but the same type of recombination is going to be occurring at all the autosomes in this fly and in this fly. So there's recombination occurring on other chromosomes in both of the mating types, but in this experiment, we're focusing on analyzing only the output of the W and M genes on the X chromosome due to recombination in this female. Okay, so great. Here's a depiction of what might happen. I want to point out that sometimes you see these things lined up as 4 lines indicating the 4 chromosomes, but then they always show these X's between, the 2 outside ones, or the 2 inside ones, or the outside and the 3rd one, and so on and so forth. And I just want to keep in mind that these 2-dimensional depictions don't necessarily match exactly how the chromosomes are aligning, physically inside the cell. That's something that we really don't have a very good idea of because it's a microscopic phenomenon and structural problem that we don't have a good fix on. But we know that you can have all sorts of recombination events between outside and inside chromosomes as we depict them in a 2-dimensional illustration. So I just want to keep in mind that this is a kind of artificial depiction, not necessarily how these 4 things are configured in the cell when they line up in the synaptonemal complex. Okay, the book goes through a whole bunch of possible recombinant variations. What it does is it starts with A plus B plus chromosome and the AB chromosome. It creates a synaptonemal complex between these 2 types of chromosomes, and then it goes through a series of pretend examples here, 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 where you get various possible recombination events And the idea, I think, is just to show you that sometimes you could get half recombinant, half parental. Sometimes you get all recombinants. Sometimes you might have two recombination events which kind of reverse one another so that you maintain a all parental configuration. Each example is just saying that you can get different outputs in each meiosis depending on exactly what's recombining with what in that particular meiosis. I don't think it's useful to go through every one of these. You can look at it yourself. It's a little small, but you can see that you get from all these possible recombination events a variety of meiotic, haploid products that are going to be distributed to the gametes. Okay. So now a question arises, will recombination occur between two linked genes separating those alleles? Will it happen? Will it not happen? Okay? And it turns out it depends on how far apart the genes are, which is sensible. Meiotic recombination is a random process, right? We don't think of it as picking a particular spot deliberately, mostly just it happens. And the idea of it happens is going to depend on kind of sequence space. So if you have, let's say, a chromosome which has, if you have a chromosome which has big A, big B, big C, and it's a heterozygote, so the other chromosome has small a, small b, and small c, and you duplicate them and you line them up in the synaptonemal complex, the question is, what's the likelihood we will see recombination between these sets of genes? And the likelihood is greater for the B and C genes because there's a lot more distance in here where you could have recombination occur. And the likelihood is lower for recombination between A and B just because the distance, the amount of sequence available for that to happen is smaller between A and B. So there's a difference in how many recombination events you'll see or recombinant offspring you'll see, which will depend on how far apart the genes are. And originally people made use of this because you could map genes on a chromosome based on how many recombinants you'd get in various mating experiments. And so we have a kind of a definition. Each percent recombinant group corresponds to one map unit. And later we'll look at problems where we want to try to determine for some genes like ABC how far apart they are. And we'll use this kind of percent recombinance and map unit definition to draw distances. These days we don't do this for the most part. These days we can do DNA sequencing, right? So you can find out to the level of the exact nucleotide how far apart things are. And a lot of genomes have already been sequenced, so we already know how far apart a lot of these things are at the nucleotide level. But before we could do that, this was an important way to get an idea how chromosomes are organized, how far apart genes are, really fundamental questions. Okay, what else? Nodon linkage. Oh, the total number of recombinants cannot be greater than 50%. Sometimes this is a little bit confusing, but if you imagine two genes that are located on two completely different chromosomes, we know that the way that they assort in some Mendelian fashion is if you get big A, you either get big B or you get small b, right? It's kind of 50-50. If you get small a, you either get big B or small b, another 50-50 kind of option here. So this is what happens in some Mendelian case. It turns out that's the maximum. In that case, you get 50% parental, 50% recombinant, okay? If these A and B genes are actually linked together, it's possible that you'll get a lot of recombination events in between here so that they actually appear to be unlinked. So this is a particular type of situation or problem that you might encounter. And so one of the things that you're looking at in these problems or these arrays of progeny that you would see is, do you see 50% recombinant compared to 50% parental, or do you see fewer recombinant? So here's an example, kind of another crude example. We have two genes, C and D, and the maximum number of recombinant progeny you could get would be 50%. And you might get that if these genes are really, really, really far apart. So in that case, it's likely that you're going to have recombination events that will shuffle these chromosomes in such a way that they almost seem like they were unlinked and on completely different chromosomes. That's the frequency of meiotic recombination in this particular case. But you might be able to find a marker, another gene, in between the two, such that you could say, okay, here between C and E, I can identify 25% recombinance, and between E and D, I can identify 45% recombinance. So that will tell you right away that E is farther away from D than it is from C, because you're getting more recombinance here. But it would also tell you that C and D are actually linked, right? But you couldn't tell that over here because you didn't have any intervening marker. The C and D would act in this kind of 50-50 fashion if you only measured those two. But you have something inside here that you know has to be connected to C because of the number of recombinants being less than 50%. This has to be connected to D because the number is less than 50%. That connects C and D on the same string. Okay, good. Okay, so we'll look at two types of problems involving this issue of recombination and linkage and so forth. One is a 2-point test cross, which we'll just go over briefly because it's pretty straightforward. And then we'll look at the 3-point test cross, which is the more typical problem. So in a 2-point test cross that involves recombination and involves linked genes, the idea is that we have, let's say, a organism that's A plus B plus, A plus B plus on 2 chromosomes. And we have an organism here which is homozygous recessive, A and B on chromosome A and B on the other. We generate F1 heterozygotes, and we're going to ask the question, are we going to see recombination between the A and B genes in this heterozygote? And if we are, how frequently will it occur? So we take this and we cross it against our homozygous recessive organism, which is AB/AB, indicating 2 different chromosomes both have small a, small b on them. Okay? And then we're going to look for progeny and kind of categorize them. So we could get some progeny that show up as A plus B plus. We could get some progeny that show up as AB. Or we could get some that are A plus B or B plus A. Those would be the recombinants. Okay. So question, why do we test cross against homozygous recessive? I think I raised this earlier, but test crossing against homozygous recessive means that if this organism donates A plus, you'll see it because it's dominant to A. If this organism here donates small a, you'll see it because small a will be recessive. So anything that you get from this organism will be revealed in this type of a cross. That's the advantage of this type of test cross against a homozygous recessive individual. The other thing to point out here is that we're looking for recombination that occurs between the 2 chromosomes in this organism. Do we get crossovers between A and B in this organism to shuffle the alleles? The same crossing over mechanism or procedure, whatever, is happening here too. It's just that there's no consequence. If you recombine AB with AB, no matter what happens, you'll always get AB out again. So there may be recombination events that are occurring here, just like they're occurring here, but we don't care. We're not scoring any of that. Okay, so here's a picture from the book about this A plus B plus model that we just looked at. The idea is homozygous dominant, homozygous recessive, put them together to generate F1 heterozygotes, cross the F1 heterozygote, A plus B plus, AB, where the genes are linked on the same chromosome, against the AB organism so that we can reveal the output of recombination here. And we get 2 types of progeny, A plus B plus and AB. Those would be due to this chromosome or this chromosome being transmitted intact without any recombination event, right? So those would be the parental types. A plus B plus would be dominant AB, and it would look like this. This one would be recessive, it would look like this, just like in the precursors. So these are the parentals. The recombinants are different. You've combined A plus with B or B plus with A as a result of recombination in between these A and B genes. So we get these two categories of progeny, and again, we always count up the numbers of recombinants, count up the numbers of parentals, and we can make a determination about recombinant frequency, which tells us how far apart the A and B genes are. Okay, so this is a pretty simple example and kind of illustrates this notion of recombination separating linked genes. So the classic example, the kind of classic problem for this section is a 3-point test cross type problem, and I've just taken the one that they talk about in the book, and I'll go over that and tell you some things to be aware of and kind of how to do it. But basically, a standard problem is usually one where you're considering 3 genes at once, and you'd like to use a test cross that is against homozygous recessive at all 3 of those genes to evaluate numbers of recombinant progeny, and then based on the numbers, go back and calculate things like the order of the genes and distances. So one question in these problems will be, what's the order of the genes along the chromosome? So if we talk about genes A, B, C, the possible orders for these 3 genes would be either ABC, BCA, or CAB. These are not the same thing. If you pick any one gene and compare its relative location to another gene, they're going to be different in each of these cases. So here, A and B are close to one another, and A is far from C. Here, A is close to C, but far from B. Here, A is close to C, and it's close to B with B and C separated. So make sure you keep in mind that these are 3 different arrangements of 3 genes, A, B, and C. If it's ABC or CBA, it makes no difference. So if you have 3 genes on a chromosome oriented, ABC, and you flip the whole thing, it makes no difference. That's the same thing. So ABC equals CBA, BCA equals ACB. These are grades, aren't they? CAB, ABC. CAB equals BAC. So don't worry about the inversion of the rotation of them. Excuse me, but do worry about the relative position of the genes compared to the other ones. Okay, second question is, how far apart are the genes from one another? That's something else we want to evaluate in a standard 3-point test cross problem. Okay, so I'm going to go through a simple one that's in the book, but I want to point out that there's lots of variations of this type of problem that you might encounter in chapter problems and so forth. And those might include things like, maybe they trick you and they say there's 2 linked genes and then 1 unlinked gene. So now you've got 2 genes on one chromosome and a separate chromosome for the other one. And you get all these progeny, it's confusing. So that's a variant, okay? Or maybe genes on X chromosome compared to autosomes. In that case, there might be something you have to worry about in terms of the male and female ratios being different. So that's something to consider. Or 4 genes. If it's 4 genes, usually the approach will be do 3 first and then add in the 4th, something like that. Okay, so there can be some variations of this which are sometimes straightforward and sometimes not so straightforward. But we're going to go through the basic type of problem. Okay, so simple 3-point mapping problem. We're going to consider 3 genes and we're going to ask about linkage, order, distance, things like that. Okay, so let's suppose we're setting up a cross which we think is something like this. It's A plus B plus C plus on one chromosome, ABC on the other chromosome, so it's a heterozygote. So we're going to be able to see recombination events between the various genes here, and we cross it against the homozygous recessive organism. That's a test cross. And the point is determine gene order and distance. In this example, I'll point out, it just so happens all the plus alleles are on one chromosome. There's no really good reason for that necessarily. It makes it a lot easier to look at, okay? But it could be any parental arrangement could be parental. You could have A plus, small b, C plus, and kind of the inverse on the other chromosome. That makes no difference. This just happens to be set up. So all dominant ones are here and all recessive ones are here. It's just how this problem is. We do a test cross. I think I talked about that. We don't just get this organism, we have to prepare this, as they showed in the preceding slides, starting from true breeding parents of various sorts. We breed them together to get the heterozygotes, then we have something we can use in the experiment. So it doesn't just happen, they don't just kind of crawl out and say, okay, I'm a heterozygote. All right. And the fact, here, sometimes this throws people. Everybody naturally thinks ABC has some importance, but the fact that it's written in a certain way, let's say alphabetically, is totally irrelevant. Just because it's written ABC doesn't mean the order is ABC or CBA or something like that. That's what we're trying to find out in the problem. Okay, so keep that in mind. So here's a bunch of progeny that resulted from this cross of +++/ABC genotype against this organism, okay? And when you see a standard 3-point test cross problem, the offspring come in pretty clearly defined categories. You see one group which is present most frequently, that's the parental. That's the one where no recombination occurred between the chromosomes. The chromosomes were just kind of transmitted to the gametes without any mess or anything. And so the predominant class will be parentals in this type of problem. So we've got this number of ABC and this number of +++, okay? We have a class which is the least frequent over here. The least frequent is the most unlikely types of recombination, or the most unlikely types of recombinant progeny to occur, and they have been due to 2 separate recombination events. They're due to double recombination, and the chance of that is much lower than a single recombination event. So right away, we can look for this class of progeny which occurs in the least frequent numbers, and we know those must have been the double recombinant. So this is important. And then we've got this set of progeny in the center, which are our single recombinants. These are present at about 50, these are present at about 20. Usually you try to pair them up to be common, equally frequent. That tells you that they're between the same genes, so you will look for this category as well. If you don't see something like this, then you have to start to think about some of these other possibilities, like maybe one of the genes is unlinked or something like that, because you'll see some problems where this ratio doesn't, is not given to you, it's something else, and you've got to figure that out. Okay, so pair of recombinants with the fewest numbers are assigned as the double crossovers. Pair of the progeny with the most numbers represent parental alleles. And then one way to work this problem initially is to first try to figure out what's the order of the genes A and B and C. And there might be multiple ways you could do this. One would be you just kind of see the answer in your head, but you can kind of process these through a kind of a stepwise thing. Yeah, question?
How would we get the number?
Here?
Yeah.
The numbers are almost always given to you in a problem. I mean, but you could imagine one type of problem would be you get a set of progeny, you get a set of numbers, and you have to figure something out about the linkage, the maps, and so forth. You could imagine other problems where you maybe have to work backwards, or you have to kind of, how these things are. There's always a twist in it that confuses you, that there's different ways to set it up. Yeah. But generally, in this kind of case, you're given a set of progeny. It's not something you can know, unless you know the distance between the genes already and could maybe work your way backwards. Okay, so one approach to figuring out the order here at ABC will be to set up 3 possibilities. One is ABC, another one is BCA, another one is CAB. Again, it doesn't matter ABC versus CBA, the relative position is always the same in those cases. But here we have ABC. I took the A and moved it over here to generate BCA. I took the B and I moved it over here to generate CAB. These are 3 different possible orders. They're each distinct. And what we do, at least in this particular process, is we imagine that there were double recombination events occurring between the 3 genes, in this case, and in this case. And if we imagine that, we know we should get certain types of double recombinant chromosomes as progeny. So for instance, in this case, We line up the chromosomes A, B, C. If we have double recombination, we can think of ourselves as starting here. We collect the A, we go up, we collect B plus, we go back down, we collect C. So that's one of them. And the other output of such an event would be A plus, we go down here and collect the B, we go back up here, collect C plus. And that would be, I guess that would be the top one here. Okay, so as a result of double recombination with this particular gene order, you must get these double recombinants. Okay? And then what we do is we say, well, are these double recombinants what we actually observed in the results that we were given? Okay? And so this is A plus, B, C plus. And A plus, B, no, that doesn't work. A plus, no, that doesn't work. So you can see this order must be wrong because it doesn't generate what we know should be the double recombinants in this experiment. Okay? So we move to the next one, and the next one happens to be right. Here I've pretended the order is BCA. A double recombination event here would cause us to get small b, go up and collect C plus, go down and collect A, or B plus, go down and collect C, go back up and collect A plus. And if you look at these chromosomes, they should match what we saw here, okay? And if they match, you know that this is the correct order. So this is one way to approach it. It's very methodical, you'll get the right answer. It takes a little time maybe, but you'll get the right answer with this approach. So here, as I've set it up, the order BCA generates the correct double recombinant. Okay, so that's step one. The first part of the problem is to organize the order. And again, whether you designate it as BCA or ACD doesn't make any difference. Okay, so how do we get all the recombinant progeny and parentals in the experiment? Well, if we line up these chromosomes, and if one of the chromosomes is transmitted intact, that'll be a parental. The other chromosome transmitted intact will be another parental. Those come in the highest frequency in the problem here. The double recombinants, there's two types, right? That's this one and this one. They come in the least frequent progeny categories. And then we have two types of single recombinants. One is between the B and C genes, the other is between the C and A genes. If we have single recombination between B and C, we'll generate this output, this category of recombinants. If we have a recombination event between C and A, we generate this class of recombinant progeny. And you notice the numbers are different. The numbers are different because the distance between A and C is different from the distance between C and B, right? Recombination, the number of recombinant progeny tell us how far apart the genes are. So this is kind of an illustration of what's actually happening to generate each of these categories of progeny using now the correct gene order that we just determined. And once you do a few, you'll realize they're pretty easy. You can do them pretty quick. Okay, next step is to evaluate the distance between the genes, the gene distance, okay? So we will want to evaluate the number of recombinant progeny that were due to recombination events between, let's say, B and C. And those recombinant progeny are the 22, 22, 4, and 2. So B and C is recombination between the B and C genes here, which gives us one category 22, another category 22. Both of those are due to a recombination event between B and C, plus the doubles have a recombination between B and C as well. So we add up this number plus this number and divide it by the total, and we get, in this case, 0.1 or 10% recombinance between B and C, which gives us 10 map units. So that's one of the things usually you're going for. Okay? And CNA works the same way. You look for the number of recombinant progeny that were due to a single crossover between C and A, as well as this double crossover involving CNA as well. So you add up these plus these. That tells you the number of recombination events or progeny from crossovers here. And you, in this case, you get about 20% or 21%, something like that. Okay, so great. And then we can add those together and say that, well, probably the recombination between the outside markers will be a sum of those. So now you've generated kind of pretty much what you're going for, which is gene order and map distance. And usually in these types of simple problems, you're done, you've answered the question. Okay. There's a few types of questions where you could be given something like this or like this, and you might have to work your way backwards. The question might be, well, out of 1,000 progeny, how many would you expect to be, blah, blah, blah. So you can work forwards and backwards in these things. And then you draw out the map. So there you go. A few more notes about this. The number of double crossovers ought to be the frequency of singles here times the frequency of singles here. That's what you would predict, right? It turns out the number of doubles is less than what you would predict based on the number of single crossovers. So although this problem is kind of made up because you've got perfect 500 number of progeny and so forth, in this example, the predicted number of double crossovers would be 21% times 10%, giving us some value of both events occurring at about 2%. In fact, the numbers that they've given us in the problem show double crossovers at about 1.2%. And whether or not these data are real or not real, this does reflect what really happens. The number of double crossovers is usually less than what would be predicted by multiplying single crossover frequency times single crossover frequency. And the reason is, if you have a crossover event at some location, the cell has mechanisms to try to prevent other recombination events occurring at that same spot or nearby. All right, so there's going to be a kind of suppression of nearby recombination events, and that's why the number of doubles is less than would be predicted by this value times this value. Okay. What else? Oh, counting the number of recombinant progeny is actually going to underestimate things, because it's possible for you to have, let's say, 2 recombination events between B and C. And if that happened, then you'd go you'd go plus down here somewhere, but you'd go back up again and you'd kind of restore the plus and plus. So if you have double recombination events here, or double recombination events here, which are possible, you will never see those. Those will always appear as parentals, right? So you could have different types of recombination events in here that you might not visualize. Or if you had 3, that would be equivalent to just 1, because you'd be going back down again. Okay? If you had double recombination event in between the B and C genes, you would exchange a segment of DNA in here. So you would be swapping something here with something here, right? But you will never see that in this assay, because here we're only measuring these phenotypes or these markers A and B and C. There would be DNA exchanged in such a case, but we're not observing that. Okay, so we draw out the map. Good. Okay, and again, I just remind you, you don't necessarily need to expect all the pluses on top, all the recessives on the bottom. You can see a lot of variations. And it's just important to kind of keep things straight and work slowly and carefully as you go through them, because you make all these diagrams like this, and it's easy to blow it with something and mistake a capital letter for a small or something like that. So you got to go through them kind of methodically. Okay. Good. So, like I said, people really don't do this type of 3-point test cross experiments with Drosophila and everything anymore, because the genome is done and you know exactly where things are, and you can sequence it, in a few days or a week and find out where a mutation resides and so on and so forth. But nonetheless, these ideas are still really important to think about genes located on chromosomes and separation of alleles and so forth. And so what I've got here is a kind of illustration, a cartoon picture, indicating what a parent does with their chromosomes when they transmit DNA to their offspring. Okay, so, and it involves this idea of recombination, shuffling chromosomes or shuffling alleles. So what I've got here is one parent here, which is the mom, and another parent here, which is the dad. Okay? The mom has a haploid set of genes from her mother, which is going to be maternal grandma, and the mom has a haploid set of genes from her father, which is maternal grandpa. So however many chromosomes, I've condensed them all into this line here with a bunch of genes on it, and this line here with a bunch of genes on it, right? So this is the oocyte, this is the spermatocyte, combines together to make a diploid organism with a set of maternal alleles, set of paternal alleles. Dad is the same way. He gets a set of maternal alleles from, excuse me, from the paternal grandma. He gets a set of paternal alleles from paternal grandpa. Okay, so the parents get maternal set, paternal set, maternal set, paternal set. And what they do with them in meiosis due to recombination is they mix and match the chromosomes. Maybe, maybe it doesn't happen, maybe it happens once, maybe it happens twice, maybe it happens 3 times. There's not that many meiotic recombination events that occur, but there's a few, right? And usually this is a key point in meiosis to make sure that the chromosomes are lined up and that you should proceed with gametogenesis. Okay, so things line up and then you swap once or twice, let's say, as a general rule. As a consequence, the haploid genome that's going to be given to, in this case, the oocyte from mom, will be some shuffling of the maternal alleles she has with the paternal alleles she has, which were given to her by maternal grandma and maternal grandpa. Okay, but you don't recombine very much. So you have huge stretches of maternal DNA, huge stretches of paternal DNA, huge stretches of maternal, huge stretches of maternal. The diagram maybe doesn't illustrate this, but if this is, 200 megabytes or some super long piece of DNA, and there's 1,000 genes on here, and you have 1 recombination event or 2 recombination events, then you have a lot of alleles all connected or linked to one another that were all derived from this person. And you have, let's say in this black region, 100 or whatever number of genes that come in a particular allelic form, all linked together, and they all were derived from this person. Okay, so you get these huge swaths of sequences, all of which are contributing alleles from one of those original grandparents. Okay, so interesting to think about, and the purpose of all this is, of course, to shuffle this stuff. So parents are really mixers of DNA that they got. They mix and match it, and then they deliver some mixed and matched haploid set of chromosomes to gametes to be combined in fertilization to make a zygote, and so on and so forth. And I always think this is kind of a remarkable process because what's happening here is some really fine-scale molecular recombination of DNA in this category of germ cells. And the point of it is deliberately to mix and match sequences from parents to generate new allele combinations. So this kind of really specific recombination process has a sense that it's useful to do this because over time, a million years, you want various new combinations to percolate up and so forth so that you get new advantageous combinations of alleles. So it's almost a connection between a really specific molecular recombination process that occurs in the cell with the idea that it's useful to have this happen over huge periods of time. Yeah, question. Yeah, the idea is that there is no sense of what the sequence actually is. It could be anywhere. Yeah. Yeah, question. So if something is inbred, what that means is that it's homozygous at every allele on all the chromosomes. So even if you have recombination, it would be as though you were recombining this guy. You could say this is inbred in a sense, right? So if you have recombination here, it occurs, but there's probably not going to be much genetic output as a consequence of something like that. Okay, so this is just a way to think about what's happening in terms of parents and what they're doing with the DNA that they got, how they're rearranging it to generate new combinations, and what's being given then to the offspring as a consequence of recombination. Okay, a few more things in terms of phenotypes. Let me see. There's a lot of words here. Okay, so linkage means sometimes if the linkage is such that small a is linked to small b allele, then if an individual has small a trait, they also have small b trait, right? And in some cases, the reason for that is because the genes are linked and they have not been separated by meiotic recombination, okay? So two traits, kind of following one another sometimes means that the underlying genes are linked together and the alleles aren't separated. Another reason two traits might be linked or might associate or correlate with one another is that maybe one gene causes both of those traits. We talked about that before. There could be a single underlying cause for more than one phenotype, right? But another reason why two phenotypic traits might correlate with one another is that the gene that controls one is linked very tightly to the gene that controls the other, and they always go together. It's pretty unlikely they would be separated by recombination because they're so close together. Okay. And the other point is, what about Mendel? So Mendel studied all these things like, plant height and smooth and wrinkled and yellow and green and all that. It turns out Mendel seems like he was lucky that he picked traits that were on different chromosomes. And when you get traits that are on different chromosomes, you get those 3-to-1 ratios and 9-3-3-1 type ratios, because whatever you get at one gene is sorted independently from what you might get at another gene, because the chromosomes are completely different. So in a sense, people would say that Mendel was lucky because the traits that he happened to work on were unlinked, and so they displayed these Mendelian ratios. If they had been linked, it might have been a little bit more confusing. He would have gotten recombinant type progeny at less than 50%, and he would have had maybe a worse puzzle to try to figure out. Okay, so another kind of use of this notion of linkage versus unlinkage and recombination comes in when you try to answer questions like these. And the question is, let's suppose that you have a bunch of human pedigrees and you suspect that some trait or disorder is controlled genetically, it's inherited. Maybe it's one gene, maybe it's a few genes, but you have no idea what those genes are. And the problem is, how in the world can you identify a gene or genes that is associated with a trait or disorder when you've got 20,000 genes to work with, and you can't do all this recombinant DNA and genetic engineering kind of stuff with humans? So there's a big problem in how do you find a gene that you think causes something. And there's various ways to do this, but one way is to take advantage of this idea that genes are linked to sequences that are nearby. Okay, so my point here in the second paragraph is that genes are linked to other genes, but genes are also linked to all this DNA that's kind of on this side. And if this is the gene, there's a whole bunch of DNA over here. And there's little sequence differences all along the DNA. Compare one genome to another genome. So sometimes some allele that may cause something is always or tends to be linked to specific little molecular markers or nucleotide changes that are specific to that particular genome. It's the same idea as this. If small a allele is linked to small b allele, they're transmitted together usually and they have some effect. But maybe small a causes a disorder and there's not a gene B near it, but there's just DNA sequences. So we say small a allele may be connected to some molecular marker that's nearby, some specific sequence. Okay, so this is something we can take advantage of in these family studies. So even if you don't know what gene causes a particular disease, maybe you can identify some little molecular sequence marker that does correlate with individuals who have this disease or this disorder. So what types of sequence markers? Here's some examples. Let's suppose we have a perfectly normal gene right here, and some sequences that are nearby. They could be kilobase away, or they could be really far away. But in terms of recombination frequency, they're still tightly linked. Okay? So we have a gene that's perfectly good. We have some upstream sequence, which I've just designated as a bunch of ends. It doesn't matter what they are. But here we have a GATC motif. So this is actually a site for a restriction enzyme. And here we have an A. They're just sequences. Okay? It doesn't matter what they are. But these sequences are linked to this good gene. They tend not to be recombined away through meiotic recombination. Now let's suppose that there's another gene, which is star, which will be maybe a recessive allele of this gene, which causes some trait or disorder. And the sequences upstream of this are mostly the same as for this chromosome, but you'll find a few differences. In this case, we have a difference in the restriction enzyme site, and we call this a restriction fragment length polymorphism, because a restriction enzyme will cut here but not cut here because there's a sequence change. Okay? This one is just a single nucleotide polymorphism, just some random difference between chromosomes. Happens all the time. There's millions of these. In this case, the A is associated with the good gene, the G is associated with the recessive disease-causing gene. And these sequences are close enough to this or close enough to this that they tend not to become unlinked through meiotic recombination. This tends to stay with this tends to stay with this. If you can identify any of these things, you can identify this. That's the key here. Another type of sequence difference that occurs between people and chromosomes and all that are these micro or mini satellite repeats. We've got lots of sequences in our genome that are repetitive to various lengths. So here maybe we have good gene, whatever it is, and we have nearby a little repeat of AG, 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 whatever. Okay, this is common. These things are everywhere in our genome. And let's suppose that a recessive trait-causing variant or allele of this gene tends to be associated with sequences nearby that have a little bit longer repeat. These are the types of things that people use in these, these DNA forensic type studies, right? They look at individual differences along chromosomes and see what matches parents and relatives and so on and so forth. So we've got a couple of things that vary frequently between individuals, between chromosomes, okay? And they're very specific to those genomes. And if they're close enough to a gene, they will not be generally unlinked, okay? So now, having said all that, how does it work? The basic idea is that you have identified lots and lots and lots of these little molecular markers. You don't know what they are, you don't care what they are. They're just unique differences between people at certain locations along the length of all the chromosomes. So you can follow these through PCR or sequencing, something like that, okay? And what you try to do is take individuals that have the trait from all these families that you have, and you try to ask the question, in those individuals that have the trait, do I always see a particular marker appearing? And is that marker not appearing in normal individuals? So you're looking for, let's say at some position right here, a specific sequence marker that tends to have a unique characteristic in those individuals that have the trait and is a different sequence or a different marker in all the other normal individuals, okay? If you find such a thing, it tends to suggest that that marker is located nearby whatever gene is causing that trait. Yeah, question?
Do they use the markers because they don't know what gene causes the trait?
Sorry, start over?
Do they use the markers because they don't know what gene causes the phenotype, or do they use them just because it's easier to look at the markers than to sequence the whole gene?
So a lot of times, so if you sequence the whole genome with a bunch of different people, you'll have millions of sequence differences from one genome to another genome to another genome. And you have no idea which of those million might be causative for anything. So it's not a practical approach. Even though you can get the exact genome, you still don't know in that ocean of sequence what in the world is what, okay? And you have no idea what gene to look for. And it's going to be very time consuming to try to sequence every single gene. And even in every single gene, you may see differences from all sorts of individuals. So the reason you do it is that you know you have this kind of, let's say, library of unique sequences that cover the entire genome relatively evenly. And you're just hoping to find enough situations where a marker is connected to an individual that has the trait and suggests that because the marker is linked so closely to the gene, it's never been recombined, so you've never separated them. And so therefore, maybe that's identifying that disease gene. So once you have a marker, so you might find a marker here that always, and if you're lucky, always correlated with individuals with some particular disorder, then you would say, well, what's the gene? And you'd start to look on each side and see if you can find something that made sense for that particular disorder.
So it's like a correlation, not a.
It's a correlation, yeah. And sometimes it works, and sometimes it works falsely. Sometimes it's right. It's very difficult to do. You know, pedigree sizes are small in humans, so sometimes your population of individuals isn't very big. So there's been cases where people predicted something, and particularly for these complicated traits, you know, mental disorders and so forth, where it seems like it's multi-gene. They've predicted stuff and then it's not true and so forth. So it's very difficult to do. Yeah, question? So, yeah, so I mean, it works the best if it's a single gene that clearly causes something in an absolute yes or no fashion, right? But if you have a disorder that you suspect, or a trait you suspect is caused by 10 genes or 20 genes, and they all kind of contribute a little bit, so you're combining then this one gene, one enzyme idea with a little bit of this quantitative genetics idea, it becomes a lot more complicated. Then your correlations between things are going to weaken dramatically. Okay, is that it? Yeah, that's it. Great. So I will have exam scores posted by the weekend. And I will, I guess, see you on next Tuesday. So have a good weekend.
I also don't think they're gonna mind. But I mean, I don't think they're gonna do any of ours, to be fair.
Should be right.